Thank you for the invitation to give the presentation. Today I'm going to talk about microfluidics, specifically applied to biomedical research and to medicine. So I'm going to walk first through what is microfluidics, and then go through some examples, and then just talk about what resources we have here at OSU for fabricating those. I also have some samples sitting back in the back corner there that I put that we can talk about at the break if you're interested in finding out what these look like and how to play around with them. So first, what are microfluidics? You may have heard the term. If you're not in the field, it's a bit of a confusing term. But basically, it's just a pipe that's at the micro scale. And that can really just be one dimension of the pipe is at the micron scale. But that changes a lot of the flow physics of any fluid moving through that. And so therefore, it becomes very important when we talk about how fluid behaves as you're pushing it through. And so practically, it's really just a way of making structures that we have very controlled flow, and we can control how the liquid moves around. And if we think about where the field of microfluidics has come from, it's really been kind of coming from both ends. Clinical chemistry started seeing that miniaturization led to a lot of advantages in terms of the physics of increasing resolution of their separation processes, while at the same time there's a lot of push from defense industry, from the health industry at looking at can we make healthcare and diagnostics much more portable. Defense industry is very interested in looking at taking something out onto the battlefield. That creates a lot of problems. And so if you can miniaturize it and make it something you can carry in a suitcase, that's something they were very interested in. And we kind of met a convergence of those two interests about 15 years ago, and we've been advancing it from there. So that led to this concept that we call lab on a chip. You'll see that a lot of the papers I cite come out of a journal called Lab on a Chip. This has been a very popular term. It's also been parodied quite a bit, where you have a nice picture of a Labrador retriever sitting on a potato chip. But the basic idea is that we can take individual biochemical processes for doing separations, for selecting a protein, group of proteins based on a specific property, their charge, their size, something like that. We can convert it into a flow-through separation system, so a microcapillary tube. As you take it down farther and farther, you can increase the resolution across shorter distances. And so now if we combine several of these, we can go from doing a two-dimensional gel electrophoresis blot into just having two separate steps on a continuous path of a microfluidic system. And by introducing more fluids as it goes along, we can introduce other types of separations. We can introduce PCR reactions into that. And eventually, we come up with an entire biochemistry lab being reduced down into a single chip. And so some of these are for very specific reactions. These have been taken hold in industry. Also, just in terms of jargon, it helps to know where these fit. Point of care diagnostics is a concept. By shrinking this down, it becomes transportable. And so you can take it to where it needs to be for care. Micrototal analysis is the concept that you can take a raw sample, stick it into one of these systems, and come up with your answer on the final end. So you can stick blood in straight from a patient and get the result out. High throughput analysis is if you put enough of these in parallel, then you can run thousands of these at the same time, and therefore you can do high throughput, and you can use this for drug discovery and other applications. And then this is kind of coming from the other side. From the engineering side, we had this nice acronym of MEMS. It's anachronistic now, but and then we just put bio in front of it. But MEMS is combining electrical and mechanical, and if you take it down to the micro scale, then there's a lot of interplay, and you can control things a lot more creatively. So why are we interested in this? As I said, a lot of this has been shown from biochemical point of view of miniaturization can increase certain behaviors, and that really comes down to just as you go to smaller sizes, your surface to volume ratio becomes higher, and therefore effects that are surface-based, such as transport of heat, transport of energy, get increased relative to the volume of the sample that you're interacting with, and so therefore you get much faster putting things in, taking things out. You can also use this for affinity-based separations where interaction with a surface is what really determines how much you can separate these out. The other part of this is that interacting with structures at the nano and micro scale, we can get a lot more control. We can get a lot different effects than we expected, than what we see at the larger scale. Practically, there's been a lot of showing into this faster, cheaper higher resolution, but the basic idea is we go to smaller size scales, we're using less chemicals, so therefore each test becomes cheaper. The fields become higher, so we get faster analysis and higher resolution. The concept of putting these all together in a continuous flow gives us faster automation, easier automation, and then being able to do these in parallel gives us a lot of multiplexing. Now, there are some disadvantages. It's still relatively young, so while there are commercial products, there's still a bit of a learning curve on those commercial products. And when you're talking about working with very small volumes, how do you get a very small volume into a confined space? It can be a bit of an issue. But just as an example that these exist, they are on the market. This is from Cepheid. A bit of a disclosure, I worked there a while in the 90s at the nascent stage of this technology. But the basic idea is that this cartridge not only has the microfluidics in it, it also has all of the chemistry that's required that you can take a raw sample from a patient, a cheek swab for some of these analyses they do, put it in there, it moves the fluid around from your sample, it washes it, it runs the chemistry, and you can't see it, but on the back side of it there's an optical tube that has the PCR reaction occurring, and so it can do a full collecting sample of the cells of interest or the infected cells of interest, breaking them open, collecting the DNA, then running the PCR reactions on those. It's all done inside this machine, so once you put in the sample, the user doesn't interact with it anymore, and then they get the result out on a nice graph on the computer. And so then what the company makes their money on is just that these are disposables. It's the Gillette model that you basically give the equipment away, and then as they buy the disposables, you make the money back and then some. So in terms of what applications are there, there's been a lot of miniaturization of clinical and biochemistry, and so I've been talking about that a fair amount. But there's also now a developing field in terms of manipulating cells, both for doing separation or capture of subpopulations. So you put in a raw blood sample, you want to collect the white blood cells, or you're looking for CTCs, whatever you're looking for, by selecting based on size, based on labeling, any of these differentiable markers, we can select these subpopulations. We can just collect particles as they're flowing through, and so there's been a lot of design in terms of hydrodynamic collection of particles as they're moving through these micro devices. Or we can isolate individual cells, we can co-culture cells sitting next to other types of cells of interest, and have interface between them without necessarily having them sitting on top of each other. And then finally, another area that's developing quite a bit is looking at, as Jed was talking about, when you have these physiological structures, they don't look like what we're used to, where you think of a flat surface for your cell culture, and so producing more physiologically mimicking structures from the mechanical point of view, from the chemical point of view, and from cells growing across membranes relative to each other. So just as an example, looks like it might have got cut off a little bit. This is looking at how can we control the chemistry spatially within a microfluidic device. What you're not seeing here is that there is a fair amount of fluidic connections that are going into each of these splitting points. And so what's happening is that you have the fluid is being input up here, being split apart. You can have multiple inputs at each end of this, and then they have multiple inputs coming into here. And then just by controlling the flow rates, at the micro scale, flow is very laminar. And so therefore, any flow that's, if you have two streams coming together, they will remain as two streams flowing beside each other than for diffusion across the interface, unless you introduce some sort of three-dimensional vorticity at the micro scale. And so this is just an interesting paper showing that by having all of these multiple inputs, that you can create these very interesting patterning of chemistry. And you can think about if you just put in different signals for cells, you might be able to, for example, differentiate stem cells into different types of tissue phenotypes based on what chemistry is flowing over top of them. The other interesting point with this is also based on a valving. And so by turning a valve on or off, you can change from this pattern to this pattern just in time by allowing that valving and switching of the valving to change the flow of the liquids. There's also a lot of interest for separating out cells. So this is actually some work out of OSU. out of the NSEC, the Nanoscale Science and Engineering Center, where we have a combination of physicists working on the magnetic traps for moving magnetic particles within a microfluidic system. We have the labeling, which came out of Chalmers Lab in chemical engineering, and then the encapsulation within a two-phase flow system, again, taking advantage of the laminar flow within these systems. And so the idea is that we put in a population of cells. Only some of these cells are labeled through immunolabeling on the cell surface. As they come down through here, the ones that have these immunolabels, which are magnetic, get grabbed by these little magnetic discs sitting on the surface. And by modulating the magnetic field, having it in a rotating magnetic field, it causes the cell to hop along these magnetic discs. And you can see the individual cells here hopping along. And then they get pulled into the Cepheid flow stream, which, because it's designed to have a smaller dimension, then it leads to a higher velocity, higher shear force, pulls off the cells, and then once they get down to this oil phase, then it packages them up into this nice little aqueous package that the cell is sitting inside of there. And so we've been working some on this as well, but one of the advantages of microfluidics in the laminar flow also can become an issue. If you wanted to have cells coming in and then label them with a separate flow stream, only the cells and particles that are at the interface would run into each other, and so therefore you'd only have those cells getting labeled. So we've been working, for example, on introducing three-dimensional vorticity into the flow stream so that we can have a continuous labeling process where you would be able to, again, take a more raw sample and do some sample preparation within the microfluidics. In terms of physically doing separation, there are many techniques that people have come up with. This is an example of when you flow cells into this array of traps that a cell will come in. If there's not a cell sitting in one of these traps, it has a certain probability to be contained in the trap. Once another cell gets in there, though, then the second cell coming in would generally not be trapped by those. And so, for example, this is being used for collection of cells and then culturing them over time. If I can train that fly to be my pointer. The other one comes out of our lab, which is instead of having flow in this direction, have it flowing normal to the microfluidic system, but in this case now using some of those fiber mats that Jed was talking about, we can collect cells within these, and we have them spatially isolated from other cells, and so we've done experiments looking at, for example, cluster formation and its effects of mechanically confining clusters in terms of their differentiation. Or we can look at, for example, non-adherent cells. We can hold non-adherent cells in a continuous culture and use that for studying their biology. Or we can combine these. So this is the idea of taking these traps from the previous paper, our wells from the previous paper, and then just stacking them on top of each other in 3 dimensions, with the idea being this is a side view, this is a top view. So this is the traps sitting down over top of a well, and then as the cell flows in, first we capture it, and then by applying suction through the microwell, the pores at the bottom of the microwell, we can pull it down in there and therefore have a very specific location of the cells. And so those are being written up. Another example of a mechanical interaction, as we said, cells don't grow on a flat surface, but a lot of times they do grow on fibrous structures. And if we think about what a fiber is, it's really just kind of a one-dimensional material, and one dimension plus the half dimension to give it the diameter. So by making microfabricated structures, and I believe Dr. Gallego will speak more about this to some level, but by making microfabricated structures, we can again study the cell migration behaviors. But in this case, we can engineer the surface to have very specific interactions, so we can make very sharp corners, and we have all of these capabilities at Nanotech, and then study how different cells behave on these structures. So as he was talking about with GBM, we're very interested in looking at how those migrate, the migratory potential. We initially got into this looking at for different patient samples. Can we tell how invasive a tumor is going to be based on how fast the individual patient cells migrate? But the other part of that is different metastatic tumors have different migratory behaviors. So not just how fast do they move, because the GBM actually moves slower than brain metastatic lung cells. But the lung cells, as soon as they hit any obstacle, turn around and went back the other direction. So we get different behaviors versus the GBMs. They hit another cell and they just climbed over it and kept going in the same direction. So we see lots of different behaviors that we don't see on a flat surface. We don't see necessarily with other assays. And then kind of where the field is going is looking at, can we recapitulate even more complex tissues in microfluidic systems? And so this was just from a review paper looking at a collection of different organs on chips, and eventually by taking one of these individual organs, putting it here, then having an artificial circulatory system through a microfluidic system. You can then take it to another artificial organ, and so on. And so you could have ideally a body chip where you can test the effects of drugs as they're moving through different cell phenotypes of different organs in the body. And we can introduce a lot of different functionalities. We can have membranes where we have different cells growing on the other sides of the membranes and so on. All right, so how do we make these? If you're interested in working on these, this is my greatly oversimplified concept, but we conceptualize what we want to make. We draw up some sort of microfluidic plan. This is actually where the design comes in of how do we convert the idea we want it to flow at this flow rate over these cells in a laminar flow versus non-laminar flow and so on. We convert that into a pattern or a set of patterns that we can then make masks, do photolithography in order to make the molds, and then we pour the material. So this is for prototypes, I should also stress, that when you get to the industrial scale, you're doing a lot more of the embossing or the hot embossing or the injection molding. But most of these designs can be directly translated into the hot embossing or injection molding just by taking this PDMS pouring where it becomes a solid over top of it and converting that to the injection molding. Then we can add our connections, any sort of packaging that we need, and test it out. So this is the same basic idea in terms of that PDMS molding. We start with a photolithographically patterned structure, and then we make the negative of that. PDMS starts as a liquid. We add a cross-linker. It becomes a solid, and then we can peel that off, make connections, and then bond it to another surface. So what do these look like? Basically, this is what we get out of a simple mask, where you can have two flow channels coming in. You can see it's just a two-dimensional pattern that's been extruded into the third dimension. We can round these out a bit if we want to, just by doing some creative fabrication. But then when we package it, this is the basic idea, where you punch holes through. You can then connect these with metal tubes that go through this and move your fluid around and connect it to, pardon me, connect it to your syringe pumps and so on. And just to give you an idea of what we can fabricate at the micro scale with polymers, effectively, with a little bit of work, we can fabricate any shape that you're interested in. Anything from artificial structures, so this is a micromotor, where we can make polymer micromotors. We can take nanometer patterned lines and replicate those with these micromolding techniques, optical structures, three-dimensional microfluidic structures, or we can even take, for example, naturally occurring structures. We can make a negative mold against those naturally occurring structures and then cast a polymer on top of that and back out that same type of micro and nanoscale structure. So, for example, these are for the superhydrophobic effect. So I thought I would end then just with an overview of what we have available, if this is something you're interested in. I sit out at Nanotech West, so I'm most familiar with that. We have some of these capabilities at other locations in engineering and in the physical sciences. But if you're interested in this, we can start you off at Nanotech or help you work at other locations. But we have very advanced photolithography for microfluidics. If you want to get into nano patterning, like the lines in the previous slide, we have e-beam lithography for doing very high resolution nano patterning. We can do mechanical micromilling. And if you want to have a larger scale structure, you want to have a larger scale mold. And then we have all of the mixing stations for doing the PDMS molding and getting bubble-free PDMS molds, and so on. And we have tons of characterization tools. We're also right down the street from the CMAS. In physical sciences, they also have a maskless photolithography system, so you don't have to go through the intermediate stage of fabricating a mask. So that's for polymer, especially PDMS microfluidics. We also have the capability of doing glass or silicon-based microfluidics. And then on campus, we have a lot of the injection molding and embossing in the CAPSI, the Center for Advanced Polymer and Composites Engineering. And then we have lots of other machining techniques on campus that can work down into the microscale. In terms of design and fabrication, if you're kind of an outsider and you need to talk to somebody about design and fabrication, you can start with me, or we have several other faculty in engineering that are into micro and nanofluidics that can help with the design. And I can refer you. So with that, I'll end my presentation, and if there's any questions or time.